So, well, today we have Xisca. I think I don't need to introduce her too much. So, well, the floor is yours. Thank you.
Okay, thank you very much for coming. As Eneco said, I think that most of you know me, but for those that not, I started in Imedea, I think 10 years ago, maybe, approximately. I started with the microbiology group doing my master's degree. And then later I did my doctorate in the same group. And then I moved to the Infibio group, now with the physics and working with Marco Paulin's group. Okay, so today I want to talk about the dynamics of the yeasts, an approach from spores to outbreeding. Okay, now we are working with the yeast. I know that maybe it's not the same topic that normally we talk about here, but I think that could be interesting just for you, for let you know what we are doing here. Okay, feel free to interrupt me if you consider that there are some aspects that you don't understand, so it's better for them following all the explanation. Okay, cool. First, I need to introduce Saccharomyces cerevisiae, that is one of the best studied eukaryotic model organisms. These are the yeasts where we are working on. Normally, these yeast cells vary a lot in size, and there are a lot of applications for the yeast, as most of you know, in food and chemical industries, fermentation, environmental technologies for a more sustainable environment, also healthcare industries, and of course, biological and biomedical research. So in this regard, okay, I present you the microorganisms where we are working on, that is Saccharomyces cerevisiae, the strain Xisca, that here I just take one of the plates that I normally use. If you want to just see closer, you are not gonna die to touch it. So feel free to just see. These are the strain that we are working. This is a pearl culture, and I have to say that it's very easy to work with it and very easy to just obtain this type of cultures. Okay, so this type of strain has an alternation between two phases, okay? Normally we have the haploid cells that changes to diploid, as you can see, just to double the gene inside. Okay, this is one of the main phases where stays the yeast cells. And when there is a lack of nutrients in the medium, then sporulate. This is that these diploid cells just have daughter cells that just get inside like a quite membrane cell that is called the ascus, okay? And when we have these four cells, is a tetrad, okay? Now, we normally, of course, the cells have different development strategies. And of course, they can decide to go to other side, but in that case that there is a lack of nutrients, they decide to go to sporulation. Maybe with a favorable environment with more nutrients, they can decide to go for budding or another type of developmental processes. Okay, so now let me explain a little about the canonical mating strategies for the yeast cells when they sporulate, okay? So first, we need to differentiate between inbreeding and outbreeding. What is inbreeding? When the sporulated cells, as I told you, we have different cells inside the ascus. In that case, cells that have the MAT alpha gene and the MAT A gene. These two genes are necessary to mate, okay? Normally, they stay inside of a diploid and then in the ascus, of course. And theoretically, if you haven't modified the organism, they mate, okay? That is a normal process in the nature. So when they mate in the same ascus, okay, there are different types of mating. That is the intratetrad, that is from the same ascus, and the intertetrad, that is from the same strain but different ascus. Okay? So these three different types of inbreeding. One that is the haploselfing, that is when one of the genes the methane switches to the mate alpha or in the contrary order, then the intra-tetrad mating that is in the same ascus, the mate A and the mate alpha mates, and then in the intertetrad, okay, that is different ascus but the same strain. What about the outbreeding? Is quite the same of intertetrad mating, but the thing here is that the strains are different, okay? So there is a mix between different strains, okay? So the confinement within the ascus, it's like to promote that mating between the products of the same meiotic division could be easier, no? Because if you are inside the ascus, it's more a tight environment, so it's easy and generate clones. And of course, this minimizes the outcrossing. So the outcrossing is commonly considered very extremely rare in the nature, but also necessary for the evolution. Okay, so this thought was changed when colleagues of us just discovered an ecological and surprising niche for the outbreeding of yeast, that are the guts of wasps, okay? So this is an image of Vespula, that is one of the genus that harbor the Saccharomyces cerevisiae strains, and they just discovered that Saccharomyces cerevisiae strains were present in Ensa gas, but it was a lack of association between seasons. So they stay there during all the year, okay? So I want to explain you a little more about this, because if they stay during all the seasons, they can just support different temperatures. That's why I wanted to show you that if we differentiate the wasp in 3 different parts, the head, the thorax, and the abdomen, where it's an anaerobic environment, there was a study of Kovacam, Stab, and Tyner, where they calculate, don't ask me why, but they calculate which was the temperature when the wasp departure from the nest and then the arrival, okay? And they saw that approximately in the abdomen, the temperature was approximately between 15 and 40 degrees. That's why these were the temperatures that we were testing in our experiments. Okay, so now we could see that the association between the Saccharomyces cerevisiae and the social wasps is very good, because these yeasts just can stay inside the wasp bag during, as I told you, seasons and years, and then is a good niche to create new types of strains, okay, and just promoting the evolution and of course the intraspecific and interspecific mating. That's as a reminder, as I told you before, if inside the same ASCII or different, okay? So Stefanini and colleagues in a study at, I think in 2012, just studied the probability of outcrossing in front of inbreeding, okay? And they just saw that, as you can see here, that the outcrossing probability inside the wasp, it was super higher compared with the outcrossing probability in grapes or wine, that are environments where normally the yeasts are abundant, no? So in the same study, they differentiate between 2 parts of the gut. Well, this is called the cropy. This is the gut is the final part. We call together this is the intestine, okay? And this could be this is the crop, and this is the gut. And they also evaluate if there was a presence of interspecies or interspecies hybrid, okay? Then when yeasts just mate, how was the frequency of mating doing the outcrossing instead of the inbreeding, okay? And they saw that, as you can see, the interspecies hybrid was very high in the crop and in the gut, and we could say that social wasps are Saccharomyces mating nests, okay? So, in conclusion, yeast outbreeding in this environment, of course, has to involve physicochemical factors, and of course the biological response of cells. So just a summary, okay, to focus you in the question, the main question. Okay, how can outbreeding occur inside the wasp hub? Okay, so we have to just have a lot of strange haploid cells that then can alternate to diploid, okay, available. Then they have to sporulate and just get into the ascus, okay? Then this ascus has to be broken, okay? And then the cells that have been sporulated needs to just go outside. And then these germinated spores, which are coming maybe from different strains, from different asci, then they could make you just doing the outbreeding, okay? And how the outbreeding could be inside the wasp's gut, okay? This is the main question that we wanted to know. And this is why the Aphrodisiac Good project formed, okay? The Aphrodisiac Good project is funded by the Human Frontier Science Program, okay? And the main goal is defining the factor promoting yeast mating within the insect intestines. This is a project where, in which we are collaborating with different colleagues from different sides of the world. As you can see here, we have colleagues from the University of Boston that are expertise in mathematics, then from the University of Turin that are specialized in biology, and from the University of Sydney that are specialized in chemistry. And we are from, of course, from the university, and we are we try to just give our expertise, would say, just to trying to solve or answer some of the questions by means of physics. So our main objective is just trying to describe the physical aspects of outbreeding inside the wasp's gut. Okay. So our main goal is that try to explain the dynamical process that allows spores to find each other and then make the outcrossing instead of inbreeding. Okay, now, so if we, of course, understand this type of mechanisms, we can learn more about the Saccharomyces cerevisiae evolution and the adaptation capability. And that's why we thought in three main lines of investigation, okay? We thought that this type of cells has to be when they are inside the asci, maybe they have to suffer some chemical stress, no? When they are inside the wasp gut, maybe there is some chemical stress that will interfere in that. So we propose to do some enzymatic digestion experiments, in fact, using zymolease, because we thought that maybe there is some type of chemical factor that could favor to degrade the ascus and then just get it more permeable and easy to break. Okay, we did these experiments at different temperatures and with different concentrations of zymolease. Then we applied mechanical stress, then to know about the physical and hydrodynamic forces. We applied some mixing shear movement, okay, like some similar that would be inside the wasp's gut. And then also we want to know about the forces to break the ascus. This is related with the, of course, the environmental parameters that are inside the wasp gut and the formation of multi-stain aggregates, because if we move the cells, we think that maybe they can form different clusters, okay, and aggregates that then could enhance the outbreeding, okay? For that, we use different staining methods. And this is like the experiments that we carry on. We consider all this stuff just for studying using different instruments that causes different movement, experiments again at different temperatures and velocities, and different experiments also just adding Ximoliasa to combine the chemical stress with the mechanical stress. Okay, so starting with some of the results. In the enzymatic digestion with Ximoliasa, our hypothesis is that to break the ascus, we need a combination of chemical and mechanical factors. So we started just studying the chemical factor that is an enzymatic digestion with Ximoliasa. Okay, our main objective is quantify the change of the contour of the shape of the cells. Because normally the cells started like a more rounded form, and when they are just processed with the Ximoliasa, they started to change the shape, and you can see like, well, you will see in here, but you can see a different shape, like it's more like a flower, I could say, okay? And you can know that this is working. So we did this at different temperatures, from room temperature until 40 degrees, okay? And then different concentration of Ximoliasa, from 0 to 0.1 in different ranges. Okay, why we are using Ximoliasa? This enzyme has a lot of lytic activity, and this is an enzyme that has been found in a culture of Astrobacter luteus, that is a facultatively anaerobic bacterium that was previously associated to parasitic wasps. So this is normal that this enzyme is inside maybe the wasp's gut. And also our colleagues from Turin just tried to do this experiment in situ with wasps, and they saw that it works. So using this enzyme, even is a chemical, we bought like a chemical enzyme, but it works. So which are the main studies that we did? Well, we started from a solid biomass, that's the plate that I show you. Then we just do liquid culture from this, okay, just taking some biomass and just put in a medium that is very, that have a lot of nutrients for the yeast. And then sporulate when we just put in another medium with a lack of nutrients, and then sporulate the cells. And they have this type of shape, okay? So then we use 96 well plates. We normally put the mix of Ximoliasa, different concentrations here with the biomass. Then we normally do in triplicate or duplicate, and then we take images, okay, under the microscope. We normally need to coat the wells, because then the cells are moving around and we cannot just take the images in the same place. And we want this because we want them to track the different shape along the time, okay, over the time. So I want to show you some of the results that we got. So fits in the blue circles, okay? Just focus on the blue circles. Here you can see, as I told you, that the shape is changing, no? It's starting more rounded and then changing. And here, they started rounding, and it seems that there is no difference, no? So, why? This is the difference between applying Ximoliasa, okay, in this case 25 micrograms per milliliter, and here not applying. So these are the differences that we normally see in cells. Okay, we have to collect a lot of data of that with the different temperatures, different Ximoliasa concentrations. Then when we have all these, just starting with the image analysis. Okay, that starts, as I told you, with the acquisition of images, then the pre-processing these images that we have to apply some filters, some masks, just to be able to work them with this. Then we did the segmentation that it's in red, because I think that it's the part that normally it's very important to do quite well, because we need to get refined contours, because then we want to calculate the difference in that. The study cannot be like a course, no? Like the contour has to be well defined. So we are working on the segmentation very hard. And then when we have this, we can do the tracking and the analysis. Okay, the tracking would be just to start putting labels to all the cells and just following that cells in the different time points that we were collecting images. Because I think that I haven't told you that we normally did the experiment during 2 hours. Okay, and just we take photos during all this time. Okay, so in brief, just to show you better some photos and you can see better, they are with, when we apply the Simoliasa treatment, these are the shapes that are before and after, okay? And then from this is information that we want to study the profiles according temperature, time, Simoliasa concentration. This is an approach that we have studied, but we are working on it. And of course, we want to calculate the interaction between Simoliasa and cells because we think that it is very interesting to know, to quantify how the degrade is the wall when we apply different concentration of Simoliasa. And of course, know if the ascus is more maybe stiff or stiffness, depending on the treatment that we are doing. So we are present to answer these questions, but the thing that we are working on is that, of course, one of these conditions, maybe the best, can be combined with the mechanical force to completely read the ascus. People say yes, but that's why I'm going to show you a little about this other part. So I told you to read the ascus and improve the outbreeding, we think that the mechanical force is needed, okay? So when we apply this mechanical force, it's like a rotational motion. That could be like the same that we have in the bath, just to digest the food. Okay, and we use different instruments for doing that, okay? And also different velocities. These are the homogenizer, the orbital shaper that we use just to give to the cells this movement. And then we collect again our samples in 1096 wet plates, taking duplicates in different time points, and then we visualize this under the microscope, okay? So, just a few images to show you that, for example, here we have the two type haploid and sporulated cells, and you can see here the cells before applying the mixing shear force that are separating between them. And then when we started to do some cycles, we can see that it is starting a clusterization between them, okay? This is a good thing because we think that as closer are the cells, then better for mate, because the Saccharomyces cerevisiae is a non-motile cell. So they need some movement just to get closer and then cooking. Okay, so I want to show you another results, maybe some specific experiment that we did with the sporulated cells using the orbital shaker at this velocity and with and without Simoliasa at 30 degrees of temperature. And here I will show you the particle size distribution, okay, as a function of the number of parameter particles in the haploid. What is it? That here we have the particle size that have depending on the number of cells per cluster. Here we can see that it's clear that there are a lot of parameter particles that are only of single cell, okay? But they started to cluster. This is for the time 4, okay? And I want to focus in this part because it seems that here there is nothing, but if we focus, we can see that there are clusters of 3, 4, or 5 cells. And with the Simoliasa is higher than without using the Simoliasa because we cannot see the blue because it's here, just behind the brown color, okay? So it's in that Simoliasa just enhances the clusterization, okay? And when we see the same for the time 11 is maybe 10 cycles more, we normally did the cycles of 20 seconds approximately, okay? 20 seconds, mixing, take sample, 20 seconds, mixing, and that's all. And we can see that here, yeah, this bar is little, okay? And they have appeared clusters of more cells, okay? 5, 6, and more, okay? So it seems that the Simoliasa helps in the clusterization. So we are also, I want just last image of this experiment, that here, if you can see maybe only on the colors, okay? Only see the colors. The orange are for the that samples without zymoliasa, and the blue ones with zymoliasa. And you can see here that first the orange was on the top. So the probability that we have clusters of one cells is higher without zymoliasa, but when we just go to the clusters of 2, 3, 4, 5, 6 cells, then the blue was on the top. What is it? That the zymoliasa theoretically enhances again the clusterization and clusters of higher density. Okay. So to finish with this part, I want to just talk about the ascus breakage. Okay, that if you can see in this image, for example, in this experiment, we can see that the ascus is completely broken. Okay, this occurs in different experiments and not for all the cells. That's why we are working on just to determine which are the specific conditions when occurs. But as you can see, it can be an interesting thing to calculate, and then of course to calculate the forces that are needed. Okay, the first approach that I have done, but and then I will explain you that we need to improve this, is that only use the different parameters that we can calculate from a cell, that it could be the area, the centroid, the circularity, eccentricity, and so on, and which parameters that are correlated, okay, that this could be this in the blue, the dark blue, here with the area. These were the parameters that I used just to differentiate between the cells, okay, the ascus and the haploid, okay, because I want to just count the ascus that have been broken, no? So I need to differentiate between the cells. And here I have a plot where we can see the percentage of broken ascus over the time. That's it. For example, here, this is up to 0. It's like that here there are more broken ascus than in the previous one, okay? So the only thing that I want to just explain to you that there are most above, no, the 0. That is that there are a high quantity of broken ascus if we compare in the different cycles. Of course, there are sometimes that not. The minus values is that there is no broken ascus from one to the other, but if we are accumulating, there is a present. But we need to improve that. That's why we're working on machine learning to train network to identify cells, okay? Because we need to just automatize better this. This could be the one image of the identification that the program does with the cells, and as you can see here, they can identify perfectly the haploid. One is an haploid, one is an ascus, and one is a cluster. So we need to just introduce this part in the speed that we are working, and just we could have more good results to show. Okay, final thing that I want to explain. Okay, I have time. I told you that again, if we increase if the cell aggregation is increasing, it could be an improvement in outbreeding, okay? Because they are close together, and then it's easy for them to make. That's why we just use different staining methods to check if the cells are closer enough, okay? We used, in that case, two different staining methods using the fluorescent, the acetate, and the milk blue. And we needed that these stains have to penetrate inside the ascus, okay? Because as I told you, the ascus just is changing, okay? They need to break the ascus, just the cells go outside, and when this occurs, we need that the fluorescence has to be persistent, okay? So these two, it seems that work quite well, okay? So the analysis that we do in that regard is that, for example, here we have 3 images. Please don't focus in that I think that is problem, but it was only a capture with the microscope. But the thing that I want to show you is that here we can see when the stains are, we have the images from the two stains, okay? Here for the Nil blue, here for the fluorescein diacetate. And here we can see the different haploid set that were staining with each fluorescence, okay? Then if we merge, okay, and here the bright field image. That's all. These are the images that we took with the microscope, okay? With one fluorescence, with the other, and the bright field. Okay, then we just check with the fluorescence, okay, in both, just checking the centers where the cells are with the high brightness. And then if we merge both results, we can see that there are some cells that have both fluorescences. In that case, this, I am only just focusing in that are just maybe making a cluster, okay? Not all the other, because here in yellow you can see the others that maybe are stained with both, okay? So if we focus only in these cells, we can just calculate the relation, just doing some quite analysis with calculating the graph with the distance within nodes and the links. And if you can see here, we are just talking at the same place, okay? These two that I showed you before, and we can just calculate that the threshold that we need to use just to say that are close enough to mate, but maybe not so close to be the same central. So we have to put the threshold, but it's very interesting that we can see that in the same, okay, they are close enough. We can see the two colors. Do you see? Because here we have the cells stained with Nil blue in blue, the cells stained with fluorescein diacetate in green, and here we can see that there are one of each. I think that's all. The conclusions that for now we can see that with the enzymatic digestion, the digestion, the shape of the cells changed. Okay, of course, also enhances and favors to break the ascus. We need to know, of course, which are the best conditions to do that. Okay, the combinations. We need to work more on it. And then we also know that the cells are clustered by mechanical force, and the ascus breakage, of course, could be from a combination of the chemical factors and the physical factors. Of course, that mixing due to the movement could promote the formation of multi-strain aggregates and then can enhance the outbreeding. And finally, well, that the biological process from spores to outbreeding results from the combination of this type of stresses. And well, we are working on the stuff that we have taught. Okay, that's all. So thank you very much for your attention. Now if you have any questions, so.
I have questions. I have several questions I will get asked from the two. So are the cells in the ascus metabolically active, and could they also release some kind of enzyme when they are mature or something like this? I don't know.
Theoretically, when they are the spores, when they are in the diploid phase, then like they are metabolically active because the daughter cells are created then conforming for cells that are inside the ascus, so they are metabolic active. And then when they are released outside the ascus also because they are sexually active, then to make to anothers. Yeah. And inside, I could say that I think that yes, but I don't think
also endogenous to destroy ascus.
Maybe yes. Maybe the colleagues that are more expertise in biological things could you understand better this thing. But I think that yes, could be some mechanism that maybe favors to breaking the ascus.
Because they cannot only rely on the presence of Archrobacter, for example, to.
Of course. of course, of course. But we don't know exactly, in fact, this is a project that just born because we don't know anything about that, and it's only an approximation, but of course, I think that could be more things involved, yeah, sure. Sure.
Okay, and what is the role of this yeast in the gut of these organisms?
Okay, what is the role? I don't know, they think that this is a good niche for them, just to stay inside. I think that for the anaerobic environment, and I think that it's also beneficial for the wasps because they help us in just digesting the food that they feed. But I don't know why they prefer this niche in comparison in another. It was very surprising, but I think that maybe it's for the environmental conditions.
In the last one.
Yes,
So about the mechanical disruptions.
yeah,
I mean, these animals, when they fly, they generate probably the wings, they generate the waves that they are, the wavelength that could help in breaking, for example. I mean, if you hear them, it's because they are generating
Yeah,
wavelength.
of course.
So waves.
this could be,
I don't know, inside the gut, you have also vibration.
yes, of course. Yeah, we started with this type of movement because we have to start with something, but we are just thinking to try different another movements. For example, more than maybe like a pump also, because we think that maybe an intestine just go little small and then big like a pump, no? And we are going to just investigating with another movements and maybe the wavelength, yeah, could be a good approach. Thank you.
Thank you very much for the talk. I really enjoyed it. Let's see, I don't know if I understood it well, but you are studying what makes better for the reproduction of this yeast inside the gut, right?
Yeah.
So, but I guess that for having higher outcrossing,
Mm-hmm.
you not only need them to reproduce, you also need to have different, I don't know, individuals, so I don't know.
Yeah,
what
different strains.
strains.
Yeah.
Because if you just have clones, then
Mhm.
they can reproduce a lot, but then you won't have higher outcrossing rates. So I was wondering, do you know where this yeast comes from? Because is it something that the animal
Normally-
has just inside, or it's
No,
getting inside?
yes.
Okay, because
Normally when the wasps just feed in some
yeah, I
fruits
think
or-
that's super cool because this insect that you put there is a pollinator,
Mhm,
and actually in the flowers, in the pollen, there is yeast.
mhm.
I don't know if this is just this exactly type of, because I don't know much about this, but I just watched once a talk of Carlos Herrera talking about the yeast in the pollen. So if this is like this, pollinators, it might be that it's not only this insect, but many pollinators that do this, and pollinators are very different in their generalization in the diet. So it might be that the role that pollinators have when visiting flowers, it changes the different types of strains that are in the gut, and then bubbles are not outcrossing. So I think that could be very cool.
Yeah, I didn't know
Check.
that. We should comment it in one meeting with our colleagues. Just thank you. It's very interesting. Yeah.
Another questions?
Well, yes, the last one. So the amount of enzyme that you use, do you think that is a natural concentration, or you are looking in the days? Because you might have a not natural break of your ascus.
Okay, good question. Normally, the concentration of Zimolias that is used to just directly extract DNA from that type of cells, it's 0.5 milligrams per milliliter. So I thought that of course, we have to just use less concentrate, less Zemolias. So the highest concentration that we use is 1 to the 5 this concentration. And I think that it could be a good concentration to work. Because we are.
The question is whether it's natural, so.
Yeah, we don't exactly know that. This is a thing that we are working, our colleagues, maybe Irene knows, I don't know.
You can comment on their experiments with the gut extract.
Ah, okay, yeah, but I don't remember the concentration that.
But they don't know, but it doesn't matter.
Exactly.
It works.
Yeah, it works. They just, our colleagues in Turin that are the person that are in charge to describe the biological process, they did some experiments with it, with the Zemoliasa concentration. I think that they use 2 or 3 of the concentration that we normally use, maybe 0.1, that is 1 to the 5th, the concentration that I told you that it's used for exactly the DNA, and it works with 0.1. But I don't know if maybe it is secretly naturally this concentration. I don't know exactly.
Can I add something that they actually did experiments directly with the gut extract to see whether there is any digestion there. It's following the same protocol that we are doing here, and it does. It does change it does digest the ascus in a similar way to the thing that we see here.
In a similar way, but it
Yeah,
doesn't
with a
mean
similar
that-
way, exactly.
It doesn't mean that it's the same enzyme.
No, it's most likely not the same enzyme.
Yes.
We don't know, they don't know what's the enzyme.
Yeah.
Yeah,
They
so, yeah, this is what actually.
Yeah, no,
Yeah,
it's
they
good,
don't know that.
but in terms of this,
Signaling, you have signaling many other bacteria and many
for
other
sure, you can.
chemical
Mhm,
things.
for sure, but they don't know.
In the end, what interests you more, what is the enzyme or what is happening in the gut, right? Whatever enzyme does it produce the same, right? It just
Exactly.
want to reproduce the same.
Exactly, the same behavior, like to see how then it works. Exactly.
Okay, so we can leave it here. Thank you very much.